Welcome back to Acing AP Biology. In this video, we will discuss the process of DNA transcription. In transcription, RNA polymerases separate the two DNA strands and attach RNA nucleotides that are complementary to the DNA template. Nucleotides are added to the 3' end. Eukaryotes have 3 different types of RNA polymerases involved in gene transcription. RNA polymerase II conducts mRNA production from DNA. RNA polymerase II only opens up DNA in the required areas, not the entire DNA molecule all at once. Specific DNA nucleotides mark where transcription should start and end. These sequences are called the initiation and termination sequences. The initiation and termination nucleotides, along with nucleotides in between, make up the transcription unit. A transcription unit includes a single gene the RNA polymerase transcribes into a single mRNA molecule that codes for a single polypeptide. Transcription consists of 3 stages: initiation, elongation, and termination. In initiation, RNA polymerase II binds to a region of DNA called the promoter, which marks the initiation site so transcription can begin. The ta-ta box is a nucleotide sequence on the promoter, which is present ahead of the initiation site. Transcription factors bind to the ta-ta box, helping show the RNA polymerase where to bind. The ta-ta box, the transcription factors, and the promoter sequence make up the transcription initiation complex. Once RNA polymerase II binds to the promoter, RNA polymerase II untwists the DNA strands and begins transcribing the DNA template into mRNA. The next stage is elongation. When transcription starts, RNA polymerase II moves down the DNA template. It untwists DNA segments around 10 nucleotides at a time. It then pairs the DNA nucleotides with complementary RNA nucleotides, adding only to the 3' end. So the mRNA molecule grows from 5' to 3'. As mRNA elongates, it pulls away from the DNA template hanging from the RNA polymerase, and the unwound DNA now goes back to its helix shape. RNA polymerase II continues moving down the DNA template, adding RNA nucleotides to the growing mRNA strand until it reaches the termination sequence. Several RNA polymerase II molecules can transcribe the same gene at the same time, allowing for the production of large numbers of that specific protein. The elongation process continues until RNA polymerase reaches the terminator sequence. The RNA polymerase II detaches and releases the complete RNA molecule. Before translation can take place, the mRNA must be processed as it leaves the nucleus. So, the mRNA molecule synthesized right after transcription is called pre-mRNA. Pre-mRNA is modified before leaving the nucleus for translation. The 5' and 3' ends are both altered. The 5' end is covalently bonded to a guanosine triphosphate cap, which protects the mRNA from degradation from hydrolytic enzymes and helps the mRNA attach to the ribosome for translation. The 3' end receives a poly-A tail, which also protects the mRNA from enzyme degradation and signals for the mRNA to exit the nucleus through the nuclear pore. The leader is a non-coding sequence of mRNA found near the 5' end. It is known as a 5' UTR. The trailer is also a non-coding sequence but is located at the 3' end and therefore is called 3' UTR. As you can see here, pre-mRNA is composed of a 5' cap, a poly A tail, the 5' UTR or the leader, the 3' UTR or the trailer, and exons and introns, which we will discuss shortly. Pre-mRNA is also modified through RNA splicing. Pre-mRNA consists of both exons and introns. Exons are sequences that are to be expressed, and introns are non-coding segments. In RNA splicing, introns are removed and exons are joined together to form mature mRNA that will leave the nucleus for translation. Introns include short sequences on the ends that indicate where RNA splicing should occur. Small nuclear ribonucleoproteins, or snRNPs, are heavily involved with RNA splicing. snRNPs, along with other proteins, make up a spliceosome, which cuts the pre-mRNA at the splicing sites on the introns, cutting them out in a loop shape. Once the introns are removed, the spliceosome splices the exons together. Ribozymes also play a role in RNA splicing. These are RNA molecules that can act as enzymes, catalyzing reactions. Introns have an important role in regulating gene expression. Introns allow for one gene to lead to the production of different proteins. This takes place because the same mRNA template can be processed differently in different types of cells. This is called alternative RNA splicing. For instance, in some templates, all the introns could be removed, and in others, some introns could be left in, leading to the synthesis of different proteins. That's it for transcription. In the next video, we will go over translation, as the mRNA is translated to a polypeptide. See you then. For more bio content, hit subscribe.